We'll get started this afternoon. Hello, my name is Dr. Rob Murphy, and I'm the Executive Director of the Havey Institute for Global Health and the John Professor of Infectious Diseases and Professor of Biomedical Engineering here at Northwestern. Thanks for taking the time to join us this afternoon for our latest installment of the Havey Institute's seminar series. Today, the topic is going to be Taenia Solium, Neurocysticercosis, an eradicable cause of epilepsy worldwide. Please take a moment to follow us on Facebook and Twitter at #FSM Global Health. In addition, we welcome you to become an Institute member to receive access to specialized resources and services that support the research infrastructure here at the Institute. For more information, you can visit our website at globalhealth.northwestern.edu/members. Today, we're really thrilled to have Dr. Hector Garcia, MD, PhD, join us today. You can see his titles up on the screen. He's Director of the Center for Global Health at Universidad Cayetano Hospital, which we have had an affiliation with for quite a while. He's head of the Cysticidal unit at the National Institute of Neurological Sciences. Oh, sorry, to translate that in English. Sorry about that. I took French, not Spanish. He's also adjunct professor at Johns Hopkins Bloomberg School of Public Health. So he's one of the foremost researchers worldwide in the study of neurocysticercosis, a preventable parasitic infection of the CNS caused by the cork tapeworm Taenia Solium. He's the primary author of 2 trendsetting studies on cysticidal treatment for neurocysticercosis, namely studying drugs such as albenzadole and praziquantel, which destroys brain cysts. Dr. Garcia's studies examined the clinical benefits of these drugs and showed that combining them increases the destruction of the brain cysts without increasing side effects, improving treatment and long-term prognosis for patients with neurocysticercosis. Dr. Garcia has a long history in global health research and training, including the successful direction of a 10-year cysticercosis elimination program in the northern coastal Peru, founded by the Bill and Melinda Gates Foundation. He leads the cysticercosis working group in Peru, a multi-institutional group working in varied aspects of cysticercosis research with a major emphasis in local capacity building. He's got over 350 peer-reviewed publications, congratulations, and a result of the research in diverse subjects of public health importance in Peru. There will be time for questions and answers. At the end, please put your questions into the Q&A box on the screen while we do this. And at this point, I'm going to turn it over to Dr. Garcia. Welcome, and thank you very much for joining us.
Thank you, Dr. Murphy, for the extensive introduction. I'm very happy to be here. Should I share my screen?
Yes.
Okay, can you see my slides now?
Yes, yes, we see them. Yeah.
Excellent. So let's go. Let's go in. Thank you for the invitation. It's a pleasure to be with you today. And during the next probably 45 minutes or so, I'm going to talk about a very interesting disease, which was always treated as a neurological disease. And it's actually an infectious disease, which generated a lot of confusion when it comes to diagnosis and particularly treatment. Before I begin, I have to say, whatever I'm presenting today, it's not personal work. We have a very large multidisciplinary, multi-institutional network in Peru that we call the Cysticidal Working Group in Peru, and includes people in very, very diverse specialties, including neurologists, neurosurgeons, epidemiologists, public health specialists, biologists, veterinarians. We even have some crazy people doing mathematical models of transmission of the disease, and other crazier people doing in silico models of molecules and antigens and looking for vaccines and diagnostics. So all of these people working together, it's what gives us an excellent frame to produce our research. You can see a picture of the group right there. And one side of this that I am particularly proud of is the capacity building component that comes with it. By now, after 20 years or more, we have approximately 30 PhD, Peruvian PhDs trained overseas, basically all of them back in Peru and developing their own research groups and expanding our reach in cysticercosis research and other diseases now.
Hector,
So
could you put it into the presentation mode on your screen?
currently, I am in the presentation mode in my screen. What are you seeing?
It's at the bottom. It's a little like kind of screen looking thing. It just makes the whole screen fill up with your slide.
No.
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 No. Okay. All right. Continue. That's okay. This will be fine.
Now I have to get back to that one. Okay. So coming back to this, we are talking about Taenia Solium cysticercosis because in endemic regions, we will get to those regions in a minute. It is cause of 30% of all seizure and epileptic disorders attributable to cysticercosis. And when you do the numbers, there's more seizures and epilepsy in developing world and developing countries. So at the end of the day, cysticercosis is the main cause of acquired epilepsy in the world. And that when we talk about acquired epilepsy, it's all epilepsies excluding genetic epilepsies and delivery hypoxia epilepsies. Most of the other causes that debuted after age 10 or 15, cysticercosis is the leading cause in the world. But also, the reason we have cysticercosis all around the world, it's because there's only one domestic animal for which the poor peasants don't need to invest money to grow it. And you guess it, it's the pig. You just have to open the door and the pig will go out and look for its food and grow for free. Unfortunately, when pigs do that, pigs have a problem. They like shit. So you can see the picture on the screen. These are 5 pigs in the highlands of Cusco in Peru. And the 5 pigs are voluntarily eating from the open sewage coming from 2 houses. And when they do that, they will close the cycle and get infected by T. solium and get cysticercosis and close the cycle. This is the WHO map of endemic areas for cysticercosis. This is WHO material, so it has to be wrong. So probably most of the gray areas are also endemic for cysticercosis. And you can see that it's not the tropical belt only. It expands to much wider areas north and south. And all of these countries, cysticercosis is present, and meta-analysis consistently shows cysticercosis as causing 30% of the epilepsies in those places. So I don't think I have to go back to this. And this is the cycle of Taenia Solium. This is the typical cycle of a complex parasite in which there are an adult living in the human intestine, shedding eggs to the environment with the stools, and then a larval population in an intermediate host. Usually the intermediate host is a herbivore and gets infected by pasturing, and the definitive host is a carnivore who eats the larvae in the pig flesh and develops the adult tapeworm. And everything will be fine if we could not have the larvae, as it happens with other tapeworms, like Taenia saginata. There's no human cysticercosis for Taenia saginata, but in T. solium, the human can be also the intermediate host and have the larval cysts anywhere in the body, particularly in the brain. A few pictures. Yeah, go ahead.
Your slides aren't advancing. Could you try to advance them one more time? I think you're probably on slide 6 now, right? We can just see the screen of the first slide. I'm sorry to interrupt.
So you are not moving from one slide to the other?
Correct, unless the rest of the audience can see something different. I only see the first slide.
Let me stop sharing and go again. How about now? What do you see?
Infected pork.
Excellent. That should be it.
Okay, perfect.
So probably what you didn't see was the map. Can you see the map now?
No, it didn't move back. It stayed on infected pork. So I don't know if there's an issue with the slide sharing.
Yep, it is. In my screen, it's moving. Let me see if I can bring it up to higher side and see if we can trick the system using this. 1 second. How about now? Are you seeing my slides?
Yeah, now they're moving. Yeah.
Yeah, now they're moving. Okay, great.
Okay, so let's try this view. I'm sorry about it. So this is the map you didn't see, where the endemic areas for cysticercosis include areas north and south of the tropical belt. And this is the cycle where the carnivore has the tapeworm and the herbivore has the larvae. And we were getting to the pictures where I was going to show a chunk of infected pork. This is the lower piece with lots of cysts. You have to take into account that this is extremely atypical. Most pigs will host less than 10 cysts in the entire pig. We will get back to that. These are the larvae after being excised from the pig, and you can see these are approximately 1 centimeter vesicles of parasitic membrane with a white dot in the interior, which represents the head of the tapeworm. So when a human host ingests pork with cysts, the head of the tapeworm will get out and look for attaching to the mucus. Let me see if I can put this to work. And I'm going to move this a little bit further. And you can see how the head of the worm gets out of the cyst and explores the environment in the intestine until it attaches from the mucus. And then it will grow a parasite hanging from the mucus with this head of a scolex, which has a double crown of hooks and 4 suckers. This extremely strong attachment is required because it will grow then a tapeworm, which is like a ribbon that may measure from 2 to 4 meters, living in the human intestine, hanging from the head. But the cystodes have developed an evolutionary advantage in which they grow in pieces or segments or proglottids. So the neck region of the worm here is the reproductive region, and these little pieces are segments that keep developing in the neck. And as the new segments develop, the older segments are more mature and get more distal. This parasite has a very rudimentary nervous system, a very interesting digestive system absorbing nutrients through the tegument, not having an intestinal tube, and a completely developed reproductive system. And it's usually a single parasite and it's hermaphroditic. So it has male and female reproductive system in each proglottid. And at some point, the parasite gets bored and has sex with itself and gets pregnant. So the final proglottid is the one you see in the right side of your screen, are actually completely filled in by the uterus of the parasite, containing 20 to 30,000 infective eggs, which are what you probably saw in one of your microbiology courses, which is this little egg containing this little embryo, the size of a red cell or slightly higher, which is what will cross the tissues of the host, be it the pig or the human, and establish as a cyst. But we physicians, we don't see aborted infections of exposure or asymptomatic infections. What we see in the hospital is symptomatic cysticercosis, which is actually the tip of the tip of the tip of the iceberg. From 1,000 people who get exposed to the parasite, probably 10 will get neurological symptoms severe enough to look for clinical care. But since there are hundreds of thousands of people or millions of people exposed to the parasite, the numbers of people with neurocysticercosis add on. And just to give you an idea, in my hospital, which is a neurological hospital in Lima, we see 1 to 2 new cases a day of neurocysticercosis, debuting with epilepsy or other neurological symptoms. And here, the presentation will depend of whether the patient has one parasite in the brain or many, whether they are alive, dying, or dead, or calcified, and whether they are large enough to cause mass effects or infiltration, and how is the immune response of the host. So there are multiple variables affecting the clinical presentation. It results in a patient with cysticercosis showing up with basically any neurological symptom, but the typical presentations are late onset epilepsy, chronic headaches, and intracranial hypertension. This is what I just said. And if we have to summarize all of this complex thing into a more simple classification, we will divide neurocysticercosis in two big boxes. When the parasites are in the brain parenchyma, they don't grow too much. They are surrounded by the parenchyma. They have mechanical limitations to grow. And they establish like cysts, survive for years, and eventually get inflamed, shrink, become an inflammatory nodule or granuloma, and then disappear or calcify. And this is the same thing I just said with pictures. And you can see the cyst here, the inflammatory nodule here, and the calcification. And we will see it better here. You can see that the brain around the cyst is quite well. There is a great homeostasis in with the parasite and the host, and minimal inflammation when the parasite is alive. There's another picture. This is two small cysts on CT, not MRI. And at some point, the host immune system will find the parasite and trigger an immune inflammatory reaction to kill it. You can see this large cyst with two major changes here. There is contrast enhancement, which means the blood-brain barrier has been broken around the parasite by the inflammation. And you can see the occipital lobe very dark, talking about edema and local inflammation. Then the parasite shrinks, becomes an inflammatory nodule, or what the literature calls enhancing lesion, and then it disappears. And apparently in 40% of the cases, it will show up as a calcified scar later. There are some rare cases in which we see thousands of little cysts, all of them inflamed. This is important to name it because here the problem is inflammation. It's like an encephalitis-like syndrome. And if you come in and use antiparasitic treatment, you will liberate more antigens, generate more inflammation, and kill your patient. So the problem here is inflammation. But also we see patients with thousands or hundreds of parasites in whom the inflammation has passed, and they have non-encephalitis, heavy cysticercosis. So when the cysts are in the parenchyma, the problem here is basically epilepsy, seizures and epilepsy and headaches. But when the parasite goes into the spaces of the brain, two bad things happen at the same time. The first thing is it grows. It grows and infiltrates. And since it's not protected by the blood-brain barrier, there is a thick inflammatory response with lots of cells in the CSF, high protein, and you may even have low glucose. And here the problem is mass effects, hydrocephalus, and still there's lots of mortality by extraparenchymal cysticercosis. You can see here a cyst in the left lateral ventricle, a cluster of cysts in the right sylvian fissure here, and the worst of the cysticercosis is when the parasite infiltrates the bases of the brain, what was called in the older literature racemose cysticercosis, because the pathology of this chronic extensive arachnoiditis resembles a bunch of grapes. And you're not seeing individual parasites here. It's just CSF or cystic fluid trapped in an extensive arachnoiditic magma of parasitic membrane. So during a couple of minutes, we run through 10 or 9 different patients. Each of them constitutes a different challenge for clinical diagnosis, serological diagnosis, radiological diagnosis, and treatment approach and prognosis. So again, if we have to summarize it, I will leave it you with parenchymal versus extraparenchymal cysticercosis. Of course, we also have cysticercosis outside the brain, And the more typical presentations are in the eye. You can see here a cyst freely floating in the vitreous of this patient. It's completely alive. And there can be also muscle calcifications where the parasite now died in an elongated form shape because it was blasted by the muscular bundles where it was dying. This is usually a casual finding and it has no major clinical consequence. Now let's talk about diagnosis of cysticercosis. My friend Oscar Del Bruno in Ecuador put together a chart of diagnostic criteria as early as in 1996. And this chart has evolved along the years, and the last version published in 2017 basically says that the diagnosis should be based in neuroimaging and supported by serological findings. Why do we say that? Because we really need to know not only whether the patient has cysticercosis, but also, as I say, the number of parasites, the size of the parasite, the location, and whether it's going to bring us a problem with antiparasitic treatment or whether do we need a surgical action now. This is supported by serology, and in serology, ELISAs for parasites in general, polyclonal ELISAs, are quite bad for antibody detection because parasites share too many epitopes and there's lots of cross reactions. So there is a test developed at CDC in 1989, which is a Western blot, which is highly sensitive and specific and detects 7 different families of antibodies. Along the time, we have learned that the number of families reflect the burden of disease, and this test is persistently seropositive for years, so it's not helpful for monitoring cure. But luckily, we now have another gadget, which is antigen ELISA detection. And this is not an ELISA for antibodies using a raw crude antigen. This is an ELISA to detect antigen using monoclonal antibodies developed against the very close parasite T. saginata. This was developed by 2 veterinarian groups in Europe, one in Edinburgh and one in Antwerp. It's not as sensitive as detecting antibody because the antigen is limited by the production of the parasite. But it's spectacular for the follow-up of patients. The levels of antigen you see there as the blue dotted line, the blue continuous line, are parallel to the area of the parasite on MRI, which is the dotted black line, and they become negative a little bit earlier than the parasite disappears in the pictures on the MRI. The blue columns represent sessions of antiparasitic treatment. In terms of molecular testing, we still don't have great tools. There is a PCR published, but it doesn't really add too much to our existing antigen and antibody detection. So the diagnosis is based on images and supported by serological tests. And now we will get in probably what is one of the most interesting stories of cysticercosis, which is how to treat neurocysticercosis. As late as the end of the 70s, there was no drug to kill the cysticercosis. All the tools the clinicians had at that time were steroids, antiepileptics, shunting, surgery to excise big lesions, or necropsy. By that time, a Mexican parasitologist, veterinarian, Dr. Clemente Robles, was dealing with pigs infected with schistosomiasis. He treated his pigs with praziquantel and found dead schistosoma and dead cysticercosis. Very smart person, Dr. Robles went to an endemic village and bought pigs with cysticercosis, gave them praziquantel, and found out that the cyst died after praziquantel. So the way I was told this history is that Dr. Robles went to his friend, the pediatrician, and told him, I have this drug that kills the parasites in the pig. And the pediatrician told him, well, we have a kid in the world with neurocysticercosis, let's give him praziquantel. It turns out that the history was more or less right, with the exception that the friend was a neurosurgeon, not a pediatrician. And they treated this kid with praziquantel. By that time, Mexico had CT scan, and they followed the evolution of the lesions. And thanks God, the kid went very well, and they published this paper in 1979, which says that now there is a drug to kill the cysticercosis, dog is dead, rabies is over. And just to give you a flavor of how much publication ethics have changed, in that paper, there is a picture of 5 centimeters of the smiling face of the kid in the last page next to the reference. So the world began to use praziquantel, but it was not an easy task. Some patients in other countries had seizures, status epilepticus, intracranial hypertension, and died during treatment. So the Brazilian group said, hey, this is inflammation. We are triggering inflammation. Let's give steroids, and steroids help to modulate that and control the side effects of the antiparasitic treatment. By the same time, another group in Mexico demonstrated that albenzadole, which was easier and cheaper than praziquantel to get, also works to kill the parasite. And some other groups saying, oh, wait, do we really need to kill this parasite? This parasite has been living in the brain for years. So people in favor of using antiparasitic treatment say we can kill it very quickly. We don't see the bad cases we see now, and patients have fewer seizures and leave less residual calcification. But there was another side of people saying, wait, this parasite has been there, the parasite is dying, so you're going to cause unnecessary brain inflammation as something you don't want in your brain, and you may even kill the patient. So the questions, major questions in the background of this strong opinionated battle of neurologists around the world was, if you kill the parasite, are you decreasing the risk of further seizures, and are you leaving any sequela? And the key word here is inflammation. This is a patient in whom we withdrew steroids too fast, and the patient didn't rebound iris-like syndrome, or encephalitis-like, and it was luckily it responded very quickly to reinstallation of IV steroids. So if we have to say what are the points that are grossly clear, is that first you have to worry about controlling the symptoms before killing the parasite. Second, if you have a large cyst, you have to do something. And the reason I'm bringing it up is because I once was called to testify in the case of a lady who was left to grow a cyst until it causes intracranial hypertension, and she died in the surgery because somebody misinterpreted the literature that said that there was not great advantage in killing parenchymal brain cysticercosis. And then you can define whether you want to use or not antiparasitic drugs depending on your individual patient. And if you are going to kill the parasites with drugs, you have to modulate the inflammatory response with steroids. Many years ago, we joined a bunch of people in Lima to see if they could agree on some concepts of antiparasitic treatment. And they agreed that if the patient had more than 5, this was arbitrary, parasites, it was probably wiser to destroy all of them at the same time rather than waiting for the natural evolution to be killing them one by one in different years and have it done under controlled conditions in a hospital. And then when we discussed granuloma, single enhancing lesion, the consensus was that there was no need for antiparasitic drugs because this parasite was already dying. Surprise, surprise, we were wrong. The evidence proven that there is a benefit of giving albendazole plus steroids. And then something that is very interesting in that paper was published in 2002 is that we recommended using neuronavigation and neuroendoscopy to take out ventricular cysticercosis. And that became very quickly the standard of care for intraventricular cysticercosis. And for the basal subarachnoid cysticercosis, no options but long-term courses of antiparasitic drugs and probably need for shunting. And if you have hydrocephalus, You have first to solve the hydrocephalus and then worry about defining your diagnosis of neurocysticercosis. So people want us to say yes or not, should we use albendazole or not? And by that time, we did a study in which we compared people with parenchymal brain cysts and have an epilepsy, everybody under anti-epileptic treatment, and half of them received antiparasitic drugs plus steroids, and half of them received a placebo comparison. And the questions were two. One is, will the cyst die in the placebo group by itself? And the second one is, if we kill the cyst, we are getting fewer seizures later. The answer to the first question was striking. 6 months later, in the placebo group, 87% of the cysts were happily alive and smiling for the picture. So they don't die, even if the patient has developed symptoms. They don't die in the short term by itself. But then when we look at albendazole, we had a 40% of surviving cysts, which was much higher than the 15% expected. The literature used to say by that time that albendazole was 85% effective. But despite this incomplete parasiticidal effect, we got much fewer partial seizures and significantly much fewer generalized seizures in this population. And I want you to look at the left side of this curve during the first 30 days, in which we got more seizures in the treated group, which is intuitive because we were attacking the parasites and generating inflammation. So by now, there is a growing body of the literature that demonstrates that if you kill the parasites, the likelihood of further seizure relapses decreases, including one of the groups that had initially published that there was no difference in clinical manifestations, but then they did reanalyze the data and found out that yes, there were fewer seizures with generalization after killing the parasites with antiparasitic drugs. And I was mentioning the studies in single enhancing lesions. The radiological results are very shaky. There's no striking difference in faster radiological resolution after antiparasitic drugs. But when you look at the clinical side of these trials, there is a very homogeneous effect on fewer seizures in people receiving albendazole and steroids for a single enhancing granuloma of the brain. So whether this is due to inflammation modulation by steroids or some sort of help from the antiparasitic drug to clear parasite remnant is unclear, but definitely there is a positive effect of using antiparasitic drugs even in this late degenerating stage. A couple of words to tell you how bad subarachnoid disease can be. You can see this is a Brazilian patient with extensive subarachnoid cysticercosis and midline deviation. But Dr. Nash at NIH demonstrated in a small series of 34 patients that if you have all the tools, you can do imaging whenever you want, you can have surgeon on hand, and you have antiparasitic drugs on lab, they obtained 0% mortality in this series. And so eventually we are looking forward to a much better prognosis of treated subarachnoid neurocysticercosis. So what will be my answer? My answer will be that in general, using antiparasitic drugs tends to be of benefit, but you have to define the patient, individualize the patient, and prioritize symptomatic management first of everything. So whether we're working in the latest years, we have demonstrated that combining two antiparasitic drugs doesn't increase side effects and increase cysticidal effects. We have seen that probably we are using too little steroids, at least during the initial weeks of treatment. And the US guidelines put together by the IDSA and ASTMH now include antiparasitic treatment as the approach of choice in most of the types of cysticercosis. So getting to the end of this talk, one thing that was in the title, and it's very important, is that cysticercosis is a major cause of epilepsy that can be eradicated. And that was written by a group of experts 30 years before. They say this can be eradicated, and cysticercosis is listed in a very short list of less than, I think there are 7 diseases with potential for eradication. So by the beginning of the 21st century, we wrote a letter to this gentleman and his wife, telling them very carefully that we could do a demonstration project to eliminate cysticercosis and develop a model. And we were confident that all the advances in technology, including pig vaccines and a drug called oxfendazole that kills all the cysts in the pig in a single dose, could help us to obtain this target. And we put together a center in a province in northern Peru, next to Ecuador. We put a laboratory, we had a CT scanner for the project, and most importantly, we hired this big, bald, fat guy in the yellow shirt, Dr. Guillermo Gonzalez from Dominican Republic. Dr. Guillermo Gonzalez is the best field epidemiologist I have seen ever working. He did a marvelous work in Tumbes. He put together a team of 180 people from the region and targeted 170 communities to test different strategies for intervention that included deparasitizing pigs, deparasitizing humans, and checking post-treatment stools to see if humans were treated from the tapeworm, and vaccinated the pigs with the Australian vaccine developed in Melbourne by Dr. Lightowers. And by the end of 7 years, we were able to expand our intervention to the whole region of Tumbes, a rural population of 80,000 people. And with the combined intervention using all the tools I just mentioned, after a year of intervention, we were able to demonstrate that there was no active transmission in 101 of 104 tested villages. And in a subset of villages that we studied in the middle of the project, 1 year after stopping the intervention, the infection had not returned in most of them. So we sustain that there is space to believe that Taenia Solium cysticercosis can be eliminated and eradicated, and that should be, for us, the pathway for controlling this disease in the following years. I saved a few slides here just in case I will not be talking by 2 p.m., and I think it's the case. Just wanted to tell you that we are working in several fronts of research, some of which are very, very interesting, particularly epileptogenesis and brain inflammation. And what we are doing is taking advantage of an in vitro model of development of the parasite that was developed in Peru in the last 10 years, in which the embryos in the eggs can be grown in vitro until they become a cyst. Unfortunately, in T. solium, we cannot get a scolex. In T. saginata, we can, but that allows us to map the transcriptomics and the proteomics and the antigen changes in the parasite, moving from the invasive embryo to the established and surviving cyst. And so we expect that that will give us more insights in diagnostics and potential treatments. But we also have a model of cysticercosis in the rat brain, in which we can get cysts with pathology similar to the human. And later, more recently, we got a model of neurocysticercosis in the pig, in which we inject the invasive oncospheres through the carotid and get consistent infection. And it's more similar to the human brain with cysticercosis. Unfortunately, we cannot get pure brain infection because pigs have this plexus in the neck. So some of the eggs divert from this and go to the neck of the pig, but most of the cysts get to the pig brain, and we can typify and those calculate to obtain pigs with 5 to 10 cysts in the whole brain, which is a perfect model to test therapeutics and anti-inflammatory models. In this model, we are trying to, we is the royal we, of course, there's many other people doing this, I'm just speaking, And we're trying to kill the parasite without leaving a calcified scar. And we have very promising data in some tools and therapies that may help us to avoid the residual calcification, which is strongly associated with seizure relapses. Finally, our collaborator Miguel Orrego is working in the cells in the subarachnoid cysticercosis membrane, which is reproductive parasite membrane, which is something completely unexpected and counterintuitive because a parasitic larvae should not grow. It's there to stay quiescent until somebody eats it and it becomes a tapeworm. So in the subarachnoid racemose membrane, Miguel has demonstrated that there is a whole population of metabolically active germinative reproductive cells that are not present in the cysts in the parenchymal variety, and more recent work is demonstrated that these cells have altered reproductive pathways, and they use the insulin pathway to grow, and they respond to metformin, and they also require iron. So he's doing a whole series of characterization of this cell population that we hope eventually will give us some insights on how to better approach subarachnoid cysticercosis and perhaps other reproductive disease caused by altered reproduction. And finally, in cysticercosis, a bunch of studies have demonstrated that cystic cells in any part of the brain can trigger hippocampal sclerosis and atrophy at distance, becoming also a cause of secondary distant epileptogenesis. That is something that we expect to study in our animal models in the very near future. So I hope not to have bored you too much during these 45 minutes, and thank you for your attention. I remain at your disposition for questions. Should I stop sharing my screen?
Yeah, that would be great, Dr. Garcia. Thank you so much for that talk. I think there's a lot of questions that I personally have about management, particularly as it relates to the global issue of epilepsy, which, as you probably know, the WHO came out a couple of years ago with a new resolution to address epilepsy. I'm going to see if there's any questions from the audience about any components of your talk.
You can feel free to put questions in the Q&A box, or you can just ask them now.
I don't see any hands raised, but I'll start by asking a question as it relates to the surgical role of managing epilepsy, particularly as it relates to parenchymal. I think neurosurgeons as a whole feel pretty comfortable navigating the intraventricular system and doing endoscopy or open craniotomies to remove cysts, calcified, whatever stage of disease the Taenia Solium is in at that point. But for parenchymal disease, what do you think is the optimal time to actually intervene, particularly in the setting of using antiparasitics and then steroids? Is it you let the brain cool off before you actually enter just because of the intense inflammatory response? Or what has been the experience of the Peruvian neurosurgeons that you work with at the Institute there?
Thank you, Roxana. That is really a lovely question. I grew in the dogma that surgery in cysticercosis was restricted to controlling hydrocephalus, excising very large lesions that were only in one place, or neuroendoscopy in the latest years. But I would say in the last 5 to 6 years, given that we have too many cases, our neurosurgeons have been treating a very interesting approach, which is to excise large lesions, even if there are other lesions that will be left untouched. And the idea here is that if you take out these large masses of parasitic membrane and fluid, you release space and you leave the brain with much less work on clearing those membranes. So what we are doing now, we again, it's the royal, we have this distortion. They are doing is they do a microcraniotomy, which even with the patient awake, that they do a microcraniotomy with a very small trepanus of 8 millimeters, and they extract the lesion and close it, usually with 1 hour 15 complete surgical time, and then send the patient for antiparasitic treatment. Another group has been trying stereotactic suction, and another group has been trying the same using neuroendoscopical exploration of the subarachnoid spaces and the peritruncal spaces to clear membranes before the patient go to antiparasitic treatment. That is completely new and very interesting and challenging. And what we use is 3 weeks as a completely arbitrary point of time to allow the brain to recover from the insult.
Yeah.
And then attack it again with antiparasitic for the remnant parasites.
Great. And is that coupled with steroids as well?
All the time. Once in a meeting, I was asked whether I would consider to treat somebody with neurocysticercosis with antiparasitic drugs without steroids. And I gave it some thought, and my answer was only if it was my mother-in-law. I really why you will want to do so.
Got it. Gina Perez asked a question, but I think you answered it. It is, when would you consider surgical resection for parenchymal disease discreetly? I think is her direct question.
Yeah, we are basically talking about lesions over 2 or 3 centimeters.
Okay. And it sounds like the next question is, could you argue that the surgical resection, the risk of inflammation would decrease and could be safer?
That is the idea behind it. But unfortunately, we only have observational cohorts because doing a controlled trial of this will be too much of a challenge and perhaps even risky.
Great.
Oh, you got to thank you.
That's good.
Great.
Good.
Any other questions from the audience? I have one more question, just in general about prevention of neurocysticercosis. What is the most effective preventive strategy? Because I know that your work in Tumbes led to, as you showed, eradication. But what within that envelope of preventive strategies actually was the most effective? And what are the challenges behind countrywide mandates to allow for that to be a sustainable act to really prevent this disease from actually propagating in populations?
Well, unfortunately, this is an endemic parasite. So if you have it there, and even if you kill all the tapeworms in one shot of massive antiparasitic treatment of the human population, eventually it will come back. So we have this harsh discussion on whether control really makes any sense or we have to eliminate. Mathematical modeling of transmission is very clear. One tapeworm carrier, it's an enormous source of infection for pigs and for humans. And to make it worse, it's not easy to surveil because it will infect humans that will have symptoms years later. So we don't have the picture right there. We have to wait for years to see the consequences of transmission 5 or 10 years before. So we are more in the tracks of trying to eliminate transmission and make it sustainable at the community level. But that requires a change of behavior, and people have to understand they have to invest a little bit in vaccinating their pigs or corralling their pigs, which is even much worse because then it's work and money to feed the pigs. Another approach that I hadn't mentioned here is that when we are working with Seth O'Neill from Oregon Health and Sciences University, is that we are now screening people in endemic villages using urine antigen testing. Urine is not invasive, people is very collaborative, and people with high urine are it's very predictive of they having significant burden of neurocysticercosis in their brain. And we are catching it early, and we are trying to treat it early, but of course, it's still an observational study. And we are trying hard in looking for a similar strategy to screen for tapeworms, but that is much more complicated. So far, it doesn't work in urine, it doesn't work in serum, and managing stool screening is a disaster. It's really
Great. Any other questions? Any final questions for Hugo? Hugo, I think we're questioned out here. Thank you very much. I want to thank everyone for coming today, and especially for Hugo for taking the time to share his insight and expertise today. He's certainly an expert in the field. Thank everyone for joining us and submitting your questions. And we invite you to join us next month for our monthly seminar. And Dr. Hema Magi, Senior Program Officer, Newborn Health at the Gates Foundation, will be our speaker. Again, thanks for joining us today. And for those that have registered, a recording of the webinar will be shared via email. And this concludes today's webinar. Thank you very much, Roxana, for leading the questions. And thank you again, Hugo, for taking the time to speak to our group.
Thank you, Dr. Murphy, and everybody. A pleasure.
Great.
Thank you, Hugo.